banned. Not only, psychedelics were banned not only for the general population, but for research also. And yet research into some of these substances, LSD, psilocybin, and so on, it showed, remarkable things. So why was it banned?
Well, it was a wonderful puzzle for all of us because the U.S. government stopped all research, about 60 projects, in one day.
Where like shit that is like very regulated in the US is like not at all out here.
I'm William Padilla Brown, citizen scientist, founder of MycoSymbiotics. In September of 2022, I was in the cloud forest of Oaxaca, Mexico, doing psilocybin research with the Entheome Foundation. So we flew out of Harrisburg, Pennsylvania, took a layover in Charlotte. We flew right out of Charlotte, right into Mexico City, got ourselves a little rental car and connected with our buddy Miguel, who showed us right over to Planton for 20. Big vibe, big political activism movement happening over in right outside the Mexican Senate. So that was really cool to see, that kind of activism around cannabis. And, we popped on in the car and Miguel actually joined us. We head down to his mom's house over in Oaxaca, 4 hours through the night, and we linked up with the crew in the morning at the hotel. From the hotel, meeting up with the crew, we all linked up and drove out to La Venta over in the Sierra Madre Sur Mountains, about 12,000 feet in elevation. We came down a super steep mountain coming into the Station 1 where we set up our accommodations, where we set up our lab. Beautiful views.
My name is Victor Angel, and I am the founder of the Entheogen Genome Project, which became the Entheome Foundation. We are on a mission to preserve all entheogenic flora, fauna, and fungi. Right now we're in San Jose del Pacifico, Oaxaca, which is the southernmost mountain range. It's one of the most diverse fungal ecosystems in the world, and it's a place that's very special for me personally.
And it was a really great place because we had really easy access to a lot of forageable area to go look for these different mushrooms, and we were able to get right into town, into San Jose del Pacifico, and interact with the mushrooms that were actively in the market so we could see what people were dealing with in comparison to what was out in the hills. The lodging was rustic. We had to maintain our own fireplaces to keep warm in the evening. I love the experiences and the expression that comes out of being out of such a conditioned reality. We spent most of the time without electricity. The electricity was going off and on. It was all powered by solar, which was kind of unfortunate for running the labs because there was a couple times where we ran into issues running the lab. We tried to run Starlink, which we had issues again with the power, which we eventually ended up connecting with a local gentleman that ran a automobile kind of fixing chop shop setup out of his plot of land. And he rigged up a car battery for us so that we could utilize that for getting some work done. So, a lot of interesting maneuvers, but I couldn't really complain about the whole situation. It was really inspiring to be out of my element. Whenever we were picking up this car battery from the chop shop gentleman, he was explaining to us that he started finding these Caesar mushrooms growing on his property during the pandemic, and him and his family didn't have enough money for food, so he said that they survived through the pandemic by eating all these Caesar mushrooms that were growing on his property. Entheome is an organization focused on doing whole genome sequencing on all psychedelic plants and fungi in the world and making it publicly accessible so individuals can learn, research, and discover all new things about these different psychedelic plants, how they produce these different compounds, and much more about their history in the world. Entheogen is derived from the Greek word entheos, meaning of God. Entheogen really means to see the God within, which I think is a really powerful concept, especially with these substances that generally take us out of our typical perspective of space and time and give us a more well-rounded understanding or connection to that feeling of oneness that connects everything that is alive and everything in the world, everything in the universe. That's why so many people, when they consume these substances, may have this feeling that they're connected with everything. I think that this is really, you know, in the root of it all, what it means, but in a more physical understanding. Entheogens are substances, chemical compounds that are known to induce hallucinogenic experiences or states of altered consciousness. Almost all entheogenic substances, entheogenic plants and fungi have some level of cultural relevancy. Through modern study, we are finding nuanced strains, nuanced mushrooms, nuanced plants that have less cultural relevancy, but many cultures all around the world have a deep history and a deep interaction with a lot of these different plants and mushrooms. And for these reasons, this cultural identity needs to be protected so that we can learn and we can understand, and also the individuals that represent that culture can maintain their heritage. On another sense, or another line of this all, in a world where 99% of all of the organisms that have ever existed on this planet no longer exist, it's very important that we protect and conserve all of the organisms we have left. And when we know that there are industries that are actively deforesting our planet, it's very important that we collect this genetic information so that we can know of this organism. Even if it's gone from our planet, we can study it from its DNA.
We are here to collect specimens, which we then sequence their DNA or their genome, which is a readout of their genetic sequence. And that enables us not only to identify them and map them properly in phylogenetic trees, but also to really understand what makes them unique, how they have been evolving, and how they relate to one another. We are here to map the ecosystem that surrounds a lot of psilocybe species in the area. We believe there's somewhere a couple dozen different psilocybe species, mainly Psilocybe zapotecorum, growing around here. So in an effort to conserve this particular species, we want to understand what grows around it in terms of other fungi, other bacteria, plants, etc., as to better protect this ecosystem. The goal for this trip was to get almost like build a dream team of mycologists, molecular biologists, people who have been in contact with different ethnobotanical traditions. We met here, set up a lab. It was quite challenging. We ran out of power. We had torrential rains for a couple of days, but we were able to accomplish more than we really expected.
I am Alan Rockefeller. I'm a mycologist. I think that the data that we're producing is really important to understand a lot of different questions. So I think it's a pretty cool project. Mushrooms are really interesting, and people really like psychedelic mushrooms. So if you're trying to get people interested in nature, then psychedelic mushrooms are a great way to do that.
A big part of the reason why we focused on psilocybin with the trip is due to the fact that the Zapotec people in Oaxaca have been historically utilizing various psilocybin species for some time. One of the founders of Entheome, Victor, is of Mexican heritage, so we were showing respects to the country and going out and to be able to illuminate information on underdescribed psilocybin species utilized by the Zapotec people. So as far as we know, there was only a few species that were being utilized, and our crew was able to go out into the field and discover underdescribed species like Psilocybe miracula. So there's many reasons why we emphasized with the psilocybin mushrooms at first. It's definitely having a moment of cultural relevancy. There's a lot of perversion of the utilization of the Sabina family image, or Maria Sabina, who was a curandera, or a healing person of this area.
These, if you're in the U.S., they call these yellowfoot chanterelles, and they sell these for a really high price at Whole Foods.
That's for real.
Yeah. But this is like the chanterelle that you get off season once our chanterelles stop, and we usually see these from the Pacific Northwest, so this is really cool to see these here.
Well, foraging in Mexico was incredible. There was a lot of different diverse terrain to get through very quickly. We could go up, we could go down, we could be in cloud forest with thick humidity. Every morning we're waking up on the mountain and there's just clouds just coming up towards us. So there was a lot of diversity, and there was mushrooms in different levels on the trees and the ground. We even had the opportunity of finding underdescribed species or an undescribed species in the Hydnum genus. So a lot of people will say, if we come across a mushroom, or if we come across a plant or any kind of organism that is not found in GenBank whenever we upload the genetic sequence, whenever we upload the barcode of that organism, and there's no reference of it in the scientific literature, a lot of people will say we found a new species. I believe using the terminology new species may be confusing to people that are not of the scientific community, because it may seem that we found something that just appeared in nature or something like this. So when we say that we find a new species, we're saying that we found a species that is undescribed or underdescribed. So we found an undescribed Hydnum species out in the forest, which was really, really cool. It's edible, so there's a lot of work that we can do with that. Alan Rockefeller got some great photographs, and I'm not sure if we got the sample for DNA barcoding, but we know where we found it, so we can go back out there again, or send somebody out there if they're out in San Jose del Pacifico. Cast of characters, we got Alan Rockefeller, genomics extraordinaire, mushroom super nerd. We had Hart Singer. He's super good with the molecular biology, and also just very, very detailed oriented when it comes to looking for these mushrooms. We had Victor. Victor's, lead with Entheome Foundation. We had Mandy Quark out there with us. We also had Chris Pauly with Tryptomics. We had Iñaki, we had Damien. And we had your boy Ian, iPsychonaut. It was a really great crew, everybody bringing a lot of different attributes to the table, so that we can have that nice stone soup. Professor Jason Slott from Ohio University gave an incredible talk where he overlapped molecular evidence of psilocybin production in mushrooms and advanced brain development in humanoids or hominids. And there was some really cool correlations. So it was kind of like the next phase of research-backed evidence around Terence McKenna's stone date theory. The goals in setting up a field lab was to be able to get the genomes or get the genetic data of these mushrooms on site. When it comes to going into different countries and interacting with mushrooms or interacting with any kind of biological specimens, there are politics around it. So it is important that if we can do the work there, to be able to do the work there, and also to be able to make that information available as quick as possible. So setting up these field labs is also helping us to hash out how to do this, because we want to be able to go deeper into these undeveloped areas, deeper into the jungle, off the grid, into the bush, away from civilization, and we want to be able to pop up these labs and have a good standard of how we operate whenever we do these field laboratories. So there isn't really a list of instructions on how to set up a field molecular biology lab. So part of all of this is figuring out how to do this kind of thing. Damien found the Psilocybe mauricula, which we were really excited about. We also had a few other different specimens that we were very interested in getting the genetic data for. So what we did is we took these samples, we took a small amount of tissue from them, we lysed the cells so that we could release the DNA from the cells. For the basic amplification for barcoding, this is really enough. We can then just add the primers and send this in for amplification in the PCR machines. We use the bento lab, we also use the mini PCR machines. And then for nanopore sequencing, we had to go a little bit further and do some purification steps with spin columns to purify a little bit of this DNA, separate it from some of the cellular material from the mushrooms to be able to run it through the nanopore. We then had to prepare a library and introduce it to the flow cell. Unfortunately, on this trip, we weren't able to go through all the way with the flow cell. There was a couple different things that inhibited us, but we were able to secure the genetic information of the different species and get those barcodes. Whole genome sequencing outside is really nuanced. To be able to get long strand purified DNA, I would have just needed a little bit more of a stable setup, a little bit more stable electricity. You don't want to be entirely in a wild environment or an outdoor environment to do whole genome sequencing. See, if we were doing barcoding, this is a little bit easier because we're only working with short little snippets of DNA. But when we're trying to get the whole genome, there are a couple of different caveats. So having a little more steady electricity, and then having a more stable environment to be working in, which could be all created in a field environment. And these are the things that we need to learn. I really wanted to figure out the logistics around shipping solar generators, because we have a lot of battery packs that we're not able to get onto the airplanes. So if we can ship some of our solar generators to the sites beforehand, then we will really have no issues with electricity. Aside from that, just having a little pop-up tent that we can utilize, or even something like our Bella Bora glove boxes that we utilize these days, can really just help me to create a clean environment anywhere that I'm at to be able to do the necessary steps for getting purified DNA for whole genome sequencing. Understanding the genome of these entheogenic substances can help us understand what environmental pressures led them to producing these compounds, which may help us understand a little bit more what their actual ecological function is. There's a lot of people that will consume these entheogenic substances that can just feel and understand what they're here for. But from a scientific understanding, this is helping us to illuminate a large swath of nature. And also in making this information publicly accessible, we can have individuals that are able to study these organisms from around the world, study their metabolic function, study how they produce these different compounds. Maybe this can help with the production of new medicines by being able to understand the synthesis of these compounds in these organisms. I think all of it just leads to a better understanding of self, really. I mean, there's a whole story of life that's been going on here on Earth, and with more pieces to the story, we can have a better understanding of how we all got here.
If we take the
Whole thing.
the outside, that's dirty.
Oh, can we maybe
The other
skin
cup.
it? Scrape it?
I tried it.
Scraping it?
It's too small.
Yeah, but what about the other cups
Yeah,
exactly?
It's a little bit bigger than the other one.
So maybe let us
We
take.
get
Are you done taking spore prints?
I guess, yeah, I have a little sample. I think that's enough.
I think it might be worth.
A little bit, and I'm surprised because white, the gills were very white. Yeah.
Okay, so yeah, I mean, if we could,
I bring it.
then that sounds like that's the play then, Jason.
Yeah.
Okay.
We can save these if we want to do metagenome.
Well,
We can use this for.
Because it'll also be interesting to see what's
I'd like
in
to
the.
do some chemo analysis of some of that,
Okay.
if possible. Like,
Yeah.
because I mean, a quantity of that amount is going to be too little for me to probably pick up on TLC.
Okay.
But we probably could get it on HPLC. Psilocin molecules could bind to each other, and norpsilocin and psilocin could probably be bound to each other. So again, creating just different polymers all the way down. It's all turtles, man. Turtles, man, all the way down.
Different like bluings to greening.
I mean, that's honestly a legit question.
It's black.
What if you could purposely bruise them and then use that to quantify and back calculate how much psilocin there was?
You want to go one more round, or do you want me to?
I'm
gonna
go for it.
Do you not open it?
It's gonna be
Say again?
dependent.
How to open it? It's a slide.
Yeah.
And you can sample dependent.
So-
I hope this
actually
gels up.
So what we're gonna do is slide it out.
Oh, it's fine. I was like trying to pop it over.
Yeah.
I'm
Yep,
afraid.
yep, yep. Do you want to leave the comb in, or do you want to take the comb out?
I live.
You're comb in? You're a comb in, man?
If you do
Okay.
an aqueous extraction, so basically if you analyze.
I don't think this is gonna
captain stem
solidify.
After it's fully dried, just using like an organic extraction like methanol.
I actually
get
have a
almost
level if we wanna
exclusively
actually try
psilocybin,
to
like just trace amounts of
Legitimate.
psilocin.
I also don't think
Yeah,
this gel is
you
gonna
go.
solidify.
Okay.
Yeah, and you get more in the cap than the stem too. We've seen that consistently. And then if you use an aqueous extraction,
Well, we didn't have
even
the,
after desiccation. Like I was under
the,
the impression that once you dry it,
protocol, so we
the
didn't know how
enzymes
much-
activated, so even if you rehydrate it after, they're
How
not
much
gonna
liquid to
react,
add?
but
Oh, no,
they
they
do.
But what about post-methanol? They are damp.
You are a nurse here.
Working against the clock, so we don't run
And
out
these
of
are
electricity.
sterile. These beads.
Yes.
Did you bring the bead?
Hey, if the electricity runs out, we'll just transport this situation somewhere else. I'm not stressing. So you think I should put this whole chunk,
There's also this
everything
chunk
in
right
there?
here.
Yeah, we should just split up the stuff.
Okay.
There's about 200 total, so that should be about right.
Milligrams?
Yep.
It's a weird freak out.
I assume your kit is optimized.
should like to use that.
Mm-hmm. I dropped a chunk off this. Is this plugged in?
Yep. Should be ready to go.
Tremors are weird.
To the pines, yeah.
If that's what it is.
I'm like, what the fuck you doing here?
No, this is just.
brought a deadly animal over.
No.
You guys want to pet it?
I do kind of want to pet it.
She's got a nice little squeak to her. I mean, she'd probably give you a bite. Watch those mandibles.
I think so.
It's 14 letters.
I was just gonna spin it with the beads in there. The DNA's not gonna be in the cells.
So, should I know your email address?
Yeah,
What's your email address, no?
I think it'll be fine.
What's your email address?
Oh
Yeah, I have magnet rack. I have a magnetic rack.
That's the thing. Do you not want to post your face with your name?
No alcohol on the table. No
Yeah,
drink.
no drinks on the table? Oh yeah, you'd be fine.
Hey Siri, start a timer for 3 minutes.
And a fluorescent dye is attached to it and is showing presence of DNA. Right here you see there's no band. That means that that sample failed. So there was not enough DNA to excite the fluorescent dye. So we're looking at that
What
empty
species is
space.
this that we're barcoding?
We're barcoding all the mushrooms that we found so far. So I have to look at my notes to tell you which species that was specifically.
I am the notes.
But yeah.
Approximately.
10 out, so approximately, according to what we ran 12. 10 of them ran, so that's about a 90% success rate.
Yeah, well we ran 11. One was a positive.
Oh, one was
control.
positive
the
control.
latter.
So
9 out of 11 passed PCR.
9 out of 11, I think that's like 82, something like that, 82%.
It's fine. And the other ones might yield results. Basically, the reason why we did this most importantly was to see that PCR worked, period.
Yep.
That the reagents worked, that the primers worked, that the extraction worked, that the equipment worked. So now we can continue doing this, and since we're going to be running a nanopore, we know that our method works, so we probably won't need to run more gels.
Look at that. Chris, where we at? Look at that shit.
Ooh,
Perfect.
That's a great amount right there.
It's a perfect amount that we need.
Yeah, almost. Is that 500 you pulled up?
It's definitely not, because I didn't let go of. Oh, it's almost 500. It's almost. That's let go all the way at 500. So there's just a little bit of air.
That's a
in
good
there.
amount recovery right there, especially with these big ass metal beads.
That's beautiful.
That's great. Delicious.
Yes, yummy.
Did we cut the tips off the?
No. These are long ass. Oh, these aren't extra longs.
Yeah, but shouldn't you cut the tips off?
Just to get
For-
so it doesn't shear.
For what reason? So it doesn't shear?
Yeah.
You think that these tips are too? I'm with whatever if you're thinking that.
I mean, that's what I do at home. I don't know. That's a Jason question.
Jason, do you think we should cut the tips off or no?
Yeah, it's probably a good idea, right?
Yeah. All right. I didn't cut the tip off on this first sample, so we'll cut the tip off the second one. What are we gonna cut the tip off with?
What do you mean by cutting off the tip?
And you don't need to cut off
Circumcision.
Just
Yeah, literally.
Yeah.
Literally.
Alcohol wipes too.
Yeah. Can we get an alcohol wipe or something?
So Jordan, can I have another bag?
Yeah.
And I'm gonna buy a beer now.
Oh, you gonna go get a beer?
Yes. One success.
I think it's more important once it precipitates to have a wide bore.
Okay. That's something that I've not ever done, so that's new to me.
Interesting.
Yeah, I've never cut the tips off.
Done it for like plant material.
And whenever when we did the work at the New York Genomic Center with Nanopore, they didn't do that.
Interesting.
Yeah.
And you still got high molecular.
Yeah, and they did high molecular.
Oh, no shit. Maybe we're just superstitious.
Mazel tov.
We were able to go from foraging, identifying a species, Psilocybe molyricula, which is usually found in high altitudes above 3,000 meters. We found one at 2,100 meters, which is a very, very uncommon sighting. Now we are in the process of extracting that DNA, putting a portion of tissue in culture so we can get very high resolution genomes out of that sample. However, we were able to barcode it here at the lab. We were also able to barcode dozens of surrounding fungi, which will give us clues as to how the ecosystem is made up. And we were also able to isolate some genetic information from Psilocybe zapotecorum, which is a very prevalent species in Mexican traditional medicine. It's also believed that it's one of the strongest psilocybe mushrooms in terms of psilocybin content. So we're really excited to take a look at that genetic sequence.
I can see there's color in it, so it definitely extracted from these mushrooms. And the mushrooms look like bruised. They look beat up a little bit. Alright, I mean we got a significant amount. We got the same amount pretty much.
All of our protocols are open. We intend to make all of our hardware open, all of our bioinformatics and software open, as to encourage people to use these same kits to produce same quality data all over the world.
Reactions for 5 minutes at room temperature, protected from light. So I need to put it somewhere dark then. Oh, this box got so beat up in this travel. Doesn't want to come out.
What does it store?
Huh? This dye that needs to stay out of the light. It's like fucking fused in there or something now. Yeah, it got wet. It doesn't matter that it got wet. That's just the box, like the
You can make a new
materials.
container for it.
Oh, I forgot it comes with their own PCR tubes. Their tubes are bigger. This is the
maybe like optical plastic too.
yeah. Theirs is the ones I'm supposed to use.
Do you want gloves to handle them so you can?
You could probably. Yeah, I think so.
You might have more.
Oh, he has more.
All right, that stuff needs to stay somewhat dark. This step I can't have the flash on.
Okay.
It'll fuck up stuff.
Gotcha.
You're like, oh, he's about to say something important for the camera.
So turn off the fucking flash. Can somebody set up the P20 to 1? I need to pull 1 microliter of the sample pretty quickly after I set this up.
This one, Rob.
I have one that will if you need to.
Got one right here for you.
Just open
Thank you.
Nice one. I was pretty happy to see this campanella yesterday. Really intricate little little mushroom that grows on twigs. I got a really good hexagonia photo. I got some craterellus and some pseudohydnum. I think pretty much everything I collected is a new species this time.
Alright, now I gotta calibrate this shit.
Hey! Was that the second one?
Yeah, but that's like nothing.
It's something.
No, it came out that way the first
Nothing,
time.
we can't work with it. But at least it's something.
PCR needs computer with Ethernet.
Yes.
Would that run locally?
It needs to do some caching.
Well, I would assume it would run locally, but we need Wi-Fi to come or probably kind of PCR software.
That we didn't get anything at all.
If we missed we messed up a step, you could have eluded it early. That's the one thing that would be the only thing that would make sense, that we extracted it, but we didn't pull it out of the column in the end. So we rinsed it out early.
So, I mean, it wouldn't hurt to try to just maybe, if we have the ability to sacrifice just a little bit of elution buffer, maybe try
I mean, I'm down to waste this whole kit.
Sure, sure.
Yeah.
So if we want to maybe try a re-elute to see if we can see if there's anything in there, is that what you were saying?
I mean, I'm down.
Yeah. Two things that we should do first then. If we maybe started with looking at whether or not what we have already worked with, maybe we didn't collect for whatever reason. We didn't handle it. We could try that. That's a quick, easy test. Just to take a small little bit and try on quanta again. The next step would probably be to try the filter. Try to elute out of the filter again.
I can grab
That
something.
might be a better working table.
Yeah, let me grab something from the room.
Event in at the cidery in upstate New York, and it was just like hash tasting. Like, it was fucking awesome. There was like so much different flavors of hash. It was like a wine tasting with hash.
Should we use your measurements or the?
I'm actually gonna start doing another column filtration DNA extraction of the Psilocybes zapotecorum. So hopefully we can get some high molecular weight DNA that we can get a whole genome from on the DNA sequencer.
So we're setting up to do a PCR on SciM that will basically amplify different exons of the gene, so we can see in Zapotec and the Psilocybe molecularum. Can't pronounce
Merlicula.
it. Merlicula. And that one will basically amplify the gene out of both and then sequence it.
Oh, that's a blast.
This is awesome.
Yeah, that's the move.
You'll need a decent amount, right?
Yeah. More the merrier for this. I'm trying to get it in. It'll be easier to take the samples or to clean it up after.
So we want to take tissue from the interior of the mushroom because we don't want contaminating organisms like bacteria and other fungi that would be on the outside. So I'm peeling open the stem and grabbing tissue from the very inside of it, which should be as clean as possible.
There we go.
I think that's enough.
6 numbers. I'll fuck around and find out. I'm gonna borrow your pipettes as well, if that's cool.
The only one this is from 9.2. That's from 9.3. This is also 9.3. The ones that you're missing here are Mandy's, and I don't know what they are.
I just have strip 7 though, so.
Did we get the power with the? All right, cool. macerating the sample into the extraction solution with a little pestle to get the DNA free from the fungal cells. See, the sample is now smooshed, mushed, mashed into the solution.
This one. It is nice, but you can't see the tubes. I guess the other ones you can't either. You can.
And I keep it cozy. I do it for the vibes.
I have to do it quietly with people having their own ceremonies.
Values, they're debatable. Reach my height if you're capable. Where I'm at is ineffable. Growing all of my vegetables. Infatuated women be pretending that they with it. They ain't with it, they just tripping. They ain't tripping when I'm tripping. Cause I'm mystic, naturalistic. With the vision and I'm handling my business.
Sí, exactamente. Can I bring it over here?
Money. Yeah, so I made the master mix for all the reactions in the centrifuge tubes. And then I'll go pipette from here into the PCR tubes with DNA. And that's 6. So you can kind of tell them apart.
What's the intention with these cordyceps? Are we supposed to eat? Are we going to eat them, or what? Are we doing science with them? Are we going to do TLC? Are we eating them, or what? I think we should do it all. I'll do TLC in my.
Like 2 a.m., 3 a.m. Okay, goodbye. So this for me is let the job at the half. So that's what I have learned. I don't know if there's other opinion.
What do you want to eat? Or I think it's fine.
Okay, we let it like this and we share.
Yeah.
And after you can just.
You probably want a lot more, right? How many people are drinking tea?
Maybe one more packet.
So we had time at the end of the trip, and Miguel said that he really wanted to see his godmother. That was a really big part of the reason why he came with us to Oaxaca, because he lives in Mexico City. It wasn't in his plans to go with us to Oaxaca, but his family was out there, so he wanted to go visit them. Miguel is a part of a multi-generational family of cannabis cultivators that have had a long history of interaction all the way back to whenever the Spanish brought cannabis to Mexico. So he brought us out into the mountains in Oaxaca to go meet his godmother because I was on a mission to find some very regional hash and traditional hash. So a nice collection of the resin produced from the cannabis grown in the mountains in Oaxaca. We walked down through a lot of different fincas on the side of this mountain, and there was just dogs everywhere. You walk on the path, you stay out of their territory, and they don't mess with you. But when we got onto, his family finca, those dogs there, we were in their territory. After a little bit of a confrontation with a German Shepherd, I left with my leg at least. But I did get a little bite on my leg on the way down. As Miguel's godmother was giving me this herbal antiseptic situation to clean out my leg, I was so caught up in the fact that I got bit by this dog that I didn't realize I was surrounded by beautiful cannabis plants. So after tending to my wounds, we were able to go inside and look at this amazing cannabis and different varieties of hash that they had been working with up there. And they even had rosin. There was some traveler that had brought a press through the mountains in Oaxaca and gave them the opportunity to press some of their mountain-grown Oaxacan cannabis into some really nice rosin.